Hello guys, welcome to the first video on this channel, and today we're going to be talking about chromosomes and mitosis. So yeah, enjoy. So by the end of this video you're going to be able to know the cell cycle and how the cell cycle is regulated. So it starts with the single sperm and egg cell when they join, and the nucleus contains 23 chromosomes, and the egg contains 23 chromosomes. So that's 46, which is what's a human body. Makes this, which is the zygote, which is the nucleus from the sperm, fuses the ovum nucleus. Now there are 46 chromosomes in the cell, 23 pairs. Millions of cells will be produced from this being the beginning. Karyotypes. Make a karyotype. The chromosomes from a dividing cell can be photographed and organized into pairs, from one of the mother and one from the father. Each pair has a characteristic size, shape, and banding pattern, except for the sex chromosomes, where the X and Y chromosomes are different. This is different types of chromosomes. Karyotype formed from a female cell, which is the X down here, and the rest of this is male, I think. Yeah, the autosomes is 22 pairs, which is all of these. The heterosome is one pair, which is this one. The 22 pairs of autosomes are known as homologous chromosomes. They have the same genes in the same places. Two pairs of homologous chromosomes, allele A, allele B, which allows them to go through. The arrows point to the corresponding genes. Visible chromosomes. When we can see distinct chromosomes, it's because the DNA molecules are tightly coiled. This is like the strands of DNA, which is normally contained in the nucleus. About 3% of one of the DNA molecules is one of the 46 chromosomes. Packing this amount into the nucleus takes some doing. That's what it does. Coil the DNA strand around histone protein, keep on coiling. So this, you can label this if you want down in the comment section, or you can I'll copy and paste a link of this so you can go through the slide for yourself and do the questions on it. So yeah, link the terms with the correct definition. I'll link this one as well. Significance of mitosis. Mitosis is the division of the nucleus between two daughter cells, and the two daughter cells have to be identical. And mitosis occurs during growth, repair of tissue, and asexual reproduction. So growth is like when you're growing up. Repair of tissues is like when you get, hurt. So you scratch your arm or your leg, and it leaves an impact on you. Asexual reproductions, sexual reproduction, same thing. Where the sperm and the egg meet, and the mitosis of the bunch of cells starts growing into a human eventually. So this is what it looked like at a very early stage. So now we need to create two identical cells. This is the homologous cell, part 1. This is 2. So yeah, this 1, 2, 1, 2. So this is the material, this is the chromosome, and this is the partner, this is the chromosomes. How does the cell produce the extra DNA? What else would the cell have to do? Can you explain the diagram? So at this point they'd have to line up. That's what we need to know. Each molecule of DNA in the nucleus replicates the produced two identical strands. The strands are called chromatids. They remain joined together in the centromere. So this is a chromatid. This is the original chromosome. This is the copy of the chromosome. So when they split, they'll have, like, this one will be 1 and this one will be 1. Like this. Sister chromatids, these are the chromatids right here, so like these. So yeah, sister. So yeah, this is the DNA molecules must replicate, so they must divide into 2 for you to grow at all. And then coil and coil again and again to make these. The number of organelles needs to increase to make you grow again. So the cell cycle is 90% interphase, is protein synthesis and growth, replication of DNA, organelles divide, DNA condenses. And then mitosis is like the bit we just talked about, which is actually not the whole cycle, is where the nuclear division is in 4 stages. Cytokinesis is where the cells divide into 2. And the cell cycle checkpoints, these control the cell type, ensure that each stage is completed properly before entering the next stage. So G1 is the cell stage, DNA synthesis complete. The G2 is cell size, DNA replication, DNA damage. M checkpoint is spindle assembly, which is the things that pulls it to the side. Pulls it to the side. And yeah. And that's all you really need to know for today. So just remember, I'll send you this link in the description. And you want to go slide 14 is what we've gone to. Make sure you like and subscribe. Be back next time for more science. Stay curious.